Good afternoon to everyone. My name is IGNACIO Negron from University of Valparaíso, uh, from the Laboratory of Cognitive Neurophysiology in Chile, and I will present the tag number 2 of the Symposium Frequency-Dependent Plasticity, the Reshaping of Oscillatory Network. This tag is entitled Task Network Frequency Specificity, Human Evidence and View from Animal Models. As introduction, it's well known that cognitive operation are supported by brain oscillations, which in human and animal models is manifested as fluctuation in the extracellular potential at several frequencies between 1 to 100 hertz. Therefore, when these cognitive operations are impaired in some pathologies, the enhancing of this oscillatory activity through brain stimulation appear as a reliable therapeutic strategy to improve cognition. However, brain stimulation generates different effects depending on the network stimulated and the frequencies of stimulation. In this symposium, we propose the task frequency specificity, in which we propose that particular neural network, as here depicted in pink and purple, implement specific frequencies for specific operation, mainly emerging from resonant properties of the network. So if you stimulate from one frequency, we can recruit one specific network. If we stimulated in another frequency, we can stimulate another different network. So the frequency of the network is specific and for some kind of behavioral task, and this because of the resonant properties of the network. In the present talk, we describe some evidence supporting this proposal, and our first evidence comes from experiment in human and performed by Andres Gomez and in the laboratory of Wael El-Derevi in the University of Valparaiso, showing the task network specificity in humans. This work is presented in more detail in the Boston number 338 of the OHBM 2022. And it's well recognized that anterior cingulate cortex is implied participates in inhibitory control of behavior. And this function is supported by theta oscillations in humans. Theta oscillation in humans is between 4 and 8 hertz. And that in older subjects it's shown an impairment in this inhibitory control. So the researcher here asked if there's impairment, this experiment is associated with theta oscillations in the anterior cingulate cortex. So to this aim, they suggested older and young participants to Simon tasks while EEG activity was recorded from the scalp. In this task, the subject must discriminate between compatible and incompatible stimulus and execute a motor response. When the cue is incompatible, the subject must to execute inhibitory control. So it's a test to evaluate inhibitory control in humans. As we can see in figure 1, they observe an increase in theta oscillations in the range of theta oscillation between 2 to 9 in the anterior cingulate cortex in young subjects when they execute inhibitory control. This is when the incongruent cue are presented. However, in older subjects, this theta is evident in slower frequency range between 2 and 6 hertz, and is also rooted in frontal and parietal cortexes when executing inhibitory control. No differences were found in the reaction time between these groups, and they also found a significant inverse correlation between the response time and the oscillatory frequency. So those subjects that showed increased response time show lower frequency for theta. As a control for they subjects these individuals to auditory recognition, and they found no difference between groups in the theta frequency range between older and young subjects. Uh, and this theta range was slower compared to incongruent to inhibitory control in young subjects. The the neural network that was recruited during this task was similar for young and older subjects. Uh, so this and they found significant correlation between behavioral response and frequency of the theta oscillations. So this also suggests that different frequencies are recruited for different tasks in healthy subjects, as we can see here, but the recruitment is impaired in all subjects, as we see here, specifically for inhibitory control, not for auditory recognition. So this activity for inhibitory control is impaired in this older subject. Indeed, they observed that the correlation between response time and theta frequency generates two different clusters for different tasks in young participant. As you can see here, in young participant is the empty squares and circles. But these clusters are less differentiated in older subjects, as you can see here, the full squares and full circles. So this suggests that the recruitment of different theta frequencies for different tasks in young subjects, as we see here, depending on the task, they can use the high theta frequency or low theta frequency. But for older subjects, didn't care if what is the test. They always for all this experimental condition, they use the same similar frequencies for all software. Indeed, we can observe that longer response time is related to less differentiation among frequencies. So this is our first evidence. The second evidence comes from optogenetic stimulation experiment in rodent models performed by Nelson Espinosa and Paulo Fuentealba from the lead team, but Paulo Fuentealba from Pontifical Catholic University in Chile, showing that neural identity supporting task network frequency specificity. Basal forebrain is a subcortical nucleus that participates in transition between internally and externally oriented brain states. Basal forebrain is conformed by at least two big nucleus, medial septum and ventral pallidum. This basal forebrain, this medial septum projects mostly to hippocampus, and ventral pallidum projects mostly to neocortex. And these state transitions modulated by basal forebrain are mediated by coupling of gamma oscillation between this basal forebrain and cortical structures. This gamma is important for this. And in this basal forebrain exists some specific GABAergic neurons called somatostatin positive neurons that support this gamma oscillation in this. Therefore, the researchers ask if exists some differential role between medial septum and ventral pallidum neurons in gamma oscillation in the basal forebrain. To this aim, they subject rodent to optogenetics inhibition of somatostatin cells, putting putting fiber optics in the medial septum and ventral pallidum to inhibit specifically somatostatin cells. And they found that this is the firing rate of somatostatin cells, specific somatostatin cells, and when they light on the laser, firing rate of some cells decreases in both nucleus, both in medial septum and ventral pallidum. So there's no difference in the inhibition of somatostatin positive cells in both nucleus. However, they observed that here that gamma oscillations were not affected in medial septum, so the inhibition of these somatostatin cells have no role in gamma oscillation in the medial septum, but in the ventral pallidum, gamma oscillation decreased when laser was delivered. Indeed, the incidence of gamma oscillation was reduced specifically in the ventral pallidum, but not in medial septum with inhibition of somatostatin cells. Also, pairwise phase consistency, a measure of phase logging of somatostatin cells with GABA oscillation, was decreased specifically in the 30 to 35 hertz in ventral pallidum, but not in medial septum. So this indicated that it's some phase locking between somatostatin cell and slow gamma oscillations in the ventral pallidum, not in the medial septum. Similarly, the power of gamma oscillation was reduced in ventral pallidum, but not in the medial septum, with optogenic resiliency of somatostatin cells. And so this suggests that somatostatin cells have a differential role for generating gamma oscillations in the ventral pallidum, but not in medial septum. So to evaluate its differential role, have behavioral manifestation. They use freely moving mice that were subject to open field to evaluate locomotor activity and alternation tasks to evaluate working memory during optogenetic inhibition of somatostatin cells. As observed in the first figure, optogenetic silencing of cells from ventral pallidum produce an increase in the speed in this open field. No effect of locomotor activity was observed when somatostatin cells in the middle septum was inhibited. Indeed, they found significant differences in speed between animals in which somatostatin cells was inhibited in ventral pallidum compared to control and compared to middle septum somatostatin cells. Contrary, inhibition of somatostatin cells in the medial septum reduce alternation task. Here is shown when the laser was on. Alternation task, a measure of working memory, was diminished in mice expressing in which inhibition was done in medial septum. But no difference were found between control mice and in animals in which the inhibition was in ventral pallidum. So these results together suggest that somatostatin cells participates in gamma oscillation in the ventral pallidum, which is involved in locomotor activity, whereas somatostatin cells in the medial septum have no participates or not participates in local gamma, but are related with working memory. And finally, our last evidence comes from optogenetic experiment in freely moving mice performed by me and my team in the University of Valparaiso, in which we test the effect of entrainment of oscillatory activity in learning. First, the prefrontal cortex supports executive aspect of learning as strategy switching or planning. We previously shown that theta and gamma oscillations emerges in the prefrontal cortex during spatial learning, which also modulates firing rate in relationship with strategy selection during navigation. And it's also well known that entrainment of brain activity disrupt naturally occurring oscillation patterns in several circuits. So we ask, is disrupting ongoing oscillations patterns in the prefrontal cortex by oscillatory entrainment affect spatial learning? To this aim, we subject freely moving mice to optogenetic entrainment of the prefrontal cortex at theta frequency to do not inhibit theta in the animals. So this is not because the absence of theta. During the execution of Barnes maze, in which it tests to evaluate spatial learning. In this test, the mice must to learn the location of an escape located always in the same place from this aversive stimulus, which is illuminated, so mice hate light. And we use optogenetic tool to entrain activity in the prefrontal cortex, but specifically during the navigation, not during the start and the goal phases of the test. So only when mice navigate in the maze, the light was on. We observed that oscillatory activity was in the prefrontal cortex was entrained in the delivered frequency of, that means 10 Hz in the mice that express channel rhodopsin, but not in control mice that do not express channel rhodopsin. This entrainment results in the significant reduction of the variability of the spectral power, as we see here. The variability decreased significantly. And also the phase resetting was significantly reduced by entrainment. Two parameters also observed in ongoing, in natural occurring oscillations in the frontal cortex. Also this variability, this entrainment decrease. This entrainment results in the significant reduction of the variability of, as we see here, the variability of the power of the oscillation, and also in a decrease of the phase resetting and an increase of theta gamma cross frequency coupling. Therefore, this optogenetical entrainment disrupts several features of naturally occurring prefrontal oscillations. Next, we evaluated behavioral performance. As we see here, the occupancy plot showed that occupancy was highly distributed in the maze in both groups during day 1, but not at day 4. This is the last day of training, in which control mice navigated near the escape, but this was not seen in general rhodopsin mice. We also found significant differences in the number of successful tries between groups and the nose-poke latency between groups, and the cumulative integrating path length is an indicative of the efficiency of the path to reach the goal. Indeed, in control mice, we observed that all these indicators are changed during training days. In this case, successful trials increase, gold nose is decreased, and CPL also decrease, indicative of efficiency of the navigation. But in channel rhodopsin expressing mice, this does not change. We also found we also evaluated in navigation strategies. Given that this parameter is indicative of executive control during learning, we evaluated navigation strategies during learning in both groups. In control group, we observed a gradual transition from less efficient random strategies to more efficient direct strategies across learning. However, this was not seen in optogenetic mice, in which channel rhodopsin was expressed. You observe that this transition is not is completely different from control mice. Indeed, the percentage of utilization of direct strategy was significantly decreased in mice expressing channel rhodopsin compared to control. So this result suggests that optogenetical entrainment, probably by disrupting ongoing oscillatory patterns, blocked spatial learning. And finally, one day after training, we evaluated memory formation. And it's important to note that during evaluation of memory, there was no entrainment. The laser was on for both groups. And we observed that in previously stimulated mice, that is channel rhodopsin mice, showed an increase in nose-poke latency, an increased number of errors, a decreased time in the goal quadrant, and an increased CPL. So compared to control. So this suggests that entrainment during training, especially during training, blocked memory formation, as non-entrainment was delivered during memory evaluation. We also finally discard permanent effects of entrainment of oscillatory activity, as we found no difference in the power of different frequencies in the prefrontal cortex. We also found no difference in variability in energy of power energy of the oscillations in phase resetting and modulation index. So this suggests that entrainment during entrainment did not produce permanent effect in oscillatory activity in the prefrontal cortex after entrainment. So, as a conclusion, our results suggest that particular oscillatory frequencies is related to a specific cognitive function in restricted neural network in young health subjects, but not in older subjects. So, is a evidence for task network frequency specificity, that this task network frequency specificity is related to the identity of neuron conforming the network, so probably the interaction among several neural population. And finally, that entrainment of oscillatory pattern is root ongoing occurring pattern which impair cognitive functions. Last, I'd like to thank to Andre Gomez and Well Alderedi from Brain Dynamics Lab, University of Valparaiso, Nelson Espinosa and Paulo Fontalva from Neural Circuitry Lab from Pontifical University Catholic University in Chile, and funding from Fondesit and Anillo from the Agencia Nacional de Investigación y Desarrollo of Government of Chile. Thank you very much.